Okay, so hi everyone and welcome to BrainMap. It's my great pleasure to introduce our speaker today, Dr. Dean Mobbs. Dean is a professor of cognitive neuroscience and Chance Scholar at the California Institute of Technology. Prior to Caltech, he was an assistant professor of psychology at Columbia University and a research assistant at Stanford University. His awards include the APS Janet Spence Award for Transformative Early Career Contributions in 2015 and the NASRAD Young Investigator Award also at 2015. He is a life fellow of Clare Hall at the University of Cambridge. I would just like to remind the audience to please address any question you have using the Q&A box. Professor Mobbs, thank you very much for coming here today. The virtual stage is all yours.
Great, thank you, and thank you for the invite to come and give a talk. It, of course, would be great to be there, but of course, COVID has stopped all of that. So, and here we are. So, what I'm going to talk about today is some of the old and some more recent research that we've been doing to try and understand the behavioral and neural basis of fear and anxiety in the human brain using brain imaging techniques. Before I start, I just want to thank all of my colleagues over the years who have played a big role in the research that I'm going to be talking about, and these different funding bodies that have supported my research. And of course, my wonderful lab. In fact, this photo is a little bit old. We were supposed to update it, but COVID happened, and actually only a couple of people in this photo in my lab still now. But actually, the research I'll be talking about will be done by some of these in this picture as well, particularly Song, Chi, Bowen, Feng. Okay. Usually on these talks, I give a background into the state of the field at the moment in terms of the theories that are driving the different research programs around the world. I'm not going to have time to do that today, but I highly recommend reading this viewpoints piece that we put together a couple of years ago now by some of the leaders in the field of affective neuroscience, and sort of pitted them together to fight out for the best theory of how to investigate and define fear. So I highly recommend reading that paper if you're interested in this topic. Sadly, because I was the moderator on this, I didn't get to talk too much about my theories, but I'll be talking about them today, and they're pretty much captured in this paper that came out about a year ago now, and in Trends in Cognitive Science, and of the same title of what today's talk will be. Okay, so the first thing we really need to do then when we're trying to understand fear and anxiety is have a solid definition of what we mean by fear and anxiety. If we go all the way back to Charles Darwin, he proposed that fear takes the gradation from mere attention to extreme terror and horror. And this is a great definition in some respects, particularly the type of research that we do, because it argues that there's a continuation, a gradation that goes from fear and potentially an anxiety. We found a bit of a solid definition of what we mean by fear and anxiety. Well, the way that we define it is that that fear is an emotion that results from the presence of an either imminent or tangible threat, while anxiety is evoked when an aversive stimulus is abstract or remote in time or space. And this is actually our clinicians, so this is not my definition, but the definition by which we abide by. And we'll go into more detail in that in a minute, and we'll talk about threat imminence continuum theory. So the way that we think about anxiety then is contextual. It's sort of bottom up, that anxiety is the context in which there's a potential to encounter a future threat. We can play around with those temporal and spatial scales associated with anxiety. And fear is when the threat is here now, okay? The tiger is in the room, for example, okay? So what I wanted to do was for when I was asking these questions during my PhD, I wanted to go back to the drawing board and try and understand the natural conditions that give rise to these different defensive responses that really sort of tap into these differences between fear and anxiety. And this link between the environment, the natural conditions, behavioral ecology, ethology is not a crazy idea, because the Nobel Prize winner Nico Tinbergen proposed that it becomes or begins to be difficult, even in some cases impossible, to say where ethology stops and neurophysiology begins. And we proposed in a paper a couple of years ago that to really understand emotions as well as the decision processes, we must consider the evolutionary and ecological conditions that give rise to them. Because if we don't, this leaves the field of affective neuroscience and decision neuroscience and the study of fear ungrounded and hard to interpret in the natural world. So we should be trying to develop paradigms that mimic the different defensive responses, mimic fear and anxiety in the natural world. That's very difficult. And really, up to this point, the standard model is really the use of one-dimensional faces, expressions, emotional expressions, or fear conditioning type experiments. And we believe that those types of paradigms don't really tap into the distinction between fear and anxiety. They can, but up to this point in time, these paradigms were not a great way to be able to dissociate fear and anxiety in the human brain. So our theoretical background then has really been based off of this idea that, first of all, we need to understand the natural conditions that give rise to different defensive responses, again, that tap into this fear and anxiety. And the natural conditions can be such things as environmental conditions. It can be such things as the traits of the threat. Is it a fast, a stealth type predator, for example, and the temporal and spatial parameters within those natural conditions, okay? And once we understand and map out those natural conditions, we can then begin to try and map onto them the organisms or the prey in this situation, the strategies, the actions and reactions that they use to evade that predator, okay? And once we can make the link between those natural conditions, okay, we can then make a link to what are the most optimal strategies that the organism can make to evade that predator, okay? And once we understand those strategies, we can then apply sort of computational models to understand how they optimize these behaviors through learning. And also we can then change the environment or the strategies by looking at how they're associated with other interconnected survival circuits, such as protection, for example. The strategies of a rodent, for example, will be different if it's protecting its offspring, or if the rodent is hungry, for example, it would become more risky. It would change its strategies within that situation. And once we understand those natural conditions and we understand what the behaviors and the strategies that they use, we can then begin to look at what happens in the brain and have a clearer understanding of how these different defensive circuits are associated with different strategic responses. Okay, and really our goal then is to try and create experiments the best that we can. It's very difficult to do realistic and natural environments in brain imaging type experiments. Of course, there's ethical principles that come to play, that we can't make people feel that they're going to die, for example, like a rodent, for example. But we can, in some respects, create conditions that mirror those real world conditions. And that's really been our goal. And our goal, and which is what we've been extending more recently for our ideas through computational ethology, is to create virtual environments where we can get more behavioral data in terms of such things as stigmataxis, which is anxiety type behavior that we see in naturalistic situations, but also in lab experiments as well, in rodents. And this is where rodents will stay around the edges of a box to protect themselves against threats. And also, can we look at things like freezing behavior and flight behavior and so on? And I'm going to talk about some of that research as we get through the talk. Okay, so one model that's been very much influential in this thinking is a model put forward by Michael Fanslow, previously known as the predatory imminence continuum or the threat imminence continuum more recently. So first of all, if we look here at the top, we have what's known as the preferred activity, the safe context. This is where our little bird, for example, is in a situation where the safest place that they can be. They spend most of their time there, okay, or would like to spend most of their time there if they're not hungry. But in this situation, the bird is in the safest place it can be, in its nest. But let's imagine now that our little bird fell out the nest onto the bed of the forest. Okay, this would be what you would call a pre-encounter defensive phase, because this is where there's an increased potential to encounter a predator. Okay, there's no predator in the environment at that point in time, but maybe our little bird here has looked down on the bed of the forest and observed a natural predator like a fox or a cat, for example. Next is the post-encounter phase, is where our little bird spots the natural predator, but there's no direct interaction between the predator and the prey. Yeah, okay. So for example, our cat is sleeping, but this is an item level of danger. Okay. And next and finally, we have the Circus Strike defensive mode. And this is where our little bird down there, you guys can see my mouse here, begins to be attacked by the cat. Okay. Now, what we see is different defensive responses under natural conditions. So a little bird here, for example, in the pre-encounter will have cautious type behaviors. You see such things as intermittent locomotion, where there'll be movements and stops, eye darting to check out the environment, avoidance of certain parts of the environment, place aversion in some respects. During post-encounter, you'll typically see freezing type responses. There's some arguments out there you also see flight. I don't want to get into that argument now, but there's some arguments out there saying that if there is a safe refuge close to, for example, a little bird here, the bird will flee towards it rather than freeze. But what you typically see here is a freezing response. And then finally, when the predator begins to attack, you'll see flight responses, escape responses. And if the threat comes too close, you'll see such things as fight or even panic type responses, where there's uncoordinated behavioral responses. So this was inspiration to us because we felt that, well, if we can create paradigms that mimic these different levels of threat imminence, the pre-post encounter and the circus strike, we can have a much better insight into what's going on in the brain during these conditions. So in a paper we published last year, we proposed that using this continuum, we can look at different defensive responses and strategies. If we move all the way over to here, in the safe context, which you can see at the top here, sorry, in A is another way of viewing Fanzo and Lester's threat imminence continuum model. So what we have is the safe. This is when the bird's in the nest, for example. And during this condition, there's no threat present. But what the animal can do is, or I should say humans in this situation, we're talking about humans here, but maybe animals can do this. We don't know. We have what we've been referring to as intermittent anxiety. This is where the animal can think about past encounters with the threat. Okay, it can ruminate about those threats. It can think about encountering those threats in the future, for example, and it's going to go into a process more of suppressing those threats because they're not a direct threat now or anywhere in the near future. But if they drop to the bed of the forest, as I mentioned during the pre-encounter threat, there's going to be this anticipatory anxiety where they could encounter the threat. And again, it's going to be this cautious behavior, this perspection, where will the threat come in the environment, these feeling states and so on. Once we get over to the post-encounter, we still have another type of anxiety, which is what we call encounter anxiety. And this is where the threat is distant, okay? But there's no attack going on, as I mentioned. But this will quickly switch over into what we refer to now as cognitive fear, okay? And this is where the threat is still not attacking you, but it's closer to you, it's more imminent, okay? So during the post-encounter phase, me and Michelle Crass, I'm not so sure if Michael agrees, but Michael Fanzo agrees, but we sort of believe, come to this conclusion independently, that the post-encounter actually is a model for anxiety and fear, the transition between these two states, okay, depending upon the proximity of the threat, despite the fact that it's not attacking you, okay. Now, cognitive fear is also in the Circus Strike attack, but this also is associated with when the threat is distant. But when the threat comes closer, when it's attacking, you see the switch over into this reactive fear systems, which we believe are in the midbrain, the paracaductal gray area. And finally, when the threat gets really close, there's no time to coordinate behavior and you get a panic type response. And we believe there's this sort of continuum based upon the threat imminence continuum, this continuum in terms of what we refer to as more of this sort of decision space or cognitive space, if you want to call it, as you get closer or as you get further away from the threat. So the further away you are from the threat, the more time you have to think, okay, the more time you have to ruminate. But as the threat comes closer, you want to start shutting down those cortical systems, hippocampus, prefrontal cortex, and so forth, and just reacting to the threat. And I'm going to come back to this when I talk about some of the empirical research. Okay, so I'm going to talk a bit now about threat imminence and human defensive circuits. And again, in that same issue in Ticks that we published, we propose that there's this interconnected set of population codes that extend from the prefrontal cortex via the amygdala and the striatum and other regions that are interconnected, such as the beta nucleus striatominalis, all the way down into the hypothalamus and the sort of more motor output systems of the ventral and lateral paraconductal gray. And our argument here is that when the threat is distant, you're going to be activating more of these strategic conscious systems. They're going to rely more on memory and prospection. And these are going to be in the ventral medial prefrontal cortex and the hippocampus. But then there'll become a switch as the threat comes closer, where there'll be more of a reactive response, where there'll be little time to be able to contemplate any strategic decisions associated with that. So the organism, in this case, we're talking about humans, always going between animals. I should really be saying that when I talk about this research, I'm talking about humans. But what we suggest is that you sort of begin to suppress these cortical regions and rely more on these reactive, if you want to use the word adwire, circuits in the midbrain. Okay. But the good thing about the population code theory is that, and we're writing a new paper about this, I'm not going to talk about this now, is that these cortical systems sort of tune themselves and learn about these threats, okay? And you go from them being these more sort of model based systems, these model-free computations that occur in the midbrain. There's this arbitration that goes on between these two systems, and work has been done in the field of reward learning, and we've been doing work more recently on this with aversive learning. So that's a little bit about the model, but I should say that also with these population codes, there's never this fully shut down between the cortex and the PAG. The animal will also begin to, as the threat begins to move further away from the threat. For example, the prey is faster than the predator. There will be this sort of dynamic interaction between these circuits, so they're never fully switched off. Okay, it's not it's not all or nothing. It's just a shift towards one system being used more than the other. Okay, so I want to give you an example now of the spatial temporal dynamics of danger and the sorts of questions that we are interested in. So some of you may have seen this. It went viral over a decade ago now, but this is a tiger. This is tourists in India, and what they're doing is they're looking down, and they know that the tiger is in that grass. Okay. So this is what you would call the post-encounter threat. Okay, there's a threat out there, but at this point they don't know that this tiger is going to attack them. So this is a good example of post-encounter. Now we're switching over to the Circus Strike. Okay, and we're seeing as the tiger gets closer and closer, you see this defensive response, and you can find out more on attackfootage.com. I should say that this individual survived with just a few injuries, minor injuries. So what we were interested in then is, of course, what goes on in the brain during post-encounter, but really in our first experiment, we wanted to see what's happening in the brain when the tiger was getting closer and closer and closer to the subject, or further and further and further away from our subject here. We're not interested in the pain itself. We're not interested in when they actually are attacked and bit or scratched and so forth. We're just interested in the difference between the spatial distance of a threat. And a good lesson is a stick is not a great weapon against a tiger. Okay, So that joke doesn't go down too well over the internet. So we created what we called an active escape task. Okay, this is a bit like Pac-Man. I'm going to brush over this quickly, where the subject is pursued in a two-dimensional maze by a virtual predator, this red dot just here. Okay, now if this red dot captures their triangle while they're being chased, again, imagine Pac-Man, they receive an electric shock. Again, we're not interested in electric shock. We remove those conditions and we have a gap between when they get caught and when the electric shock is given. We're just purely interested in the transition between these circuits in the brain associated with when the threat is close. Again, if you remember, we talked about this as cognitive fear, as these hippocampal prefrontal systems we'll talk about in a minute, to these midbrain regions, which we refer to as more as reactive fear. And again, I should clarify that when we talk about fear, we're talking about when you're under a circus strike or highly imminent threat. Okay, the predator is there in the environment. Anxiety is when there's a potential to encounter the threat in the future. But we believe there's an overlap between this cognitive fear system and the anxiety circuits in the brain, which we'll come to in a minute. So what did we find here? We found that when this red dot, this virtual predator, was further away, there was increased activity in the ventral medial prefrontal cortex. Actually, this was right there in the subgenual ACC, in the cingulate cortex just there. But as the threat comes closer to the subject, we saw that there was increased activity in the switch to this region here of the midbrain paraconductal gray. So you can see here these blobs that really surround the aqueduct of the midbrain and captures not only that region of the paraconductal gray, but also potentially colliculus as well, because the colliculus is sort of here on the outside of the midbrain. So this was a proof of concept experiment where we just wanted to see the differences between these switch between these cortical and midbrain regions and support the model that I'm not talking about now, but the Norton and Cole model that had suggested a couple of years earlier that you see this in rodents as well, as threats come closer, this switch between cortical and midbrain regions. So we proved that this in a somewhat crude experiment now, it was 2007 when we did this, and imaging has moved on quite a bit since then. But again, it proved this idea of this switch between cortical and midbrain states. So we wanted to follow this up then with a much sort of simpler task in some respects, where we wanted to look at the effects of just pure distance of, for example, in this case, a tarantula. And now how this was associated between these switches between cortical and midbrain circuits. So we created a very simple task where we put people into the MRI scanner. Okay, we put a camera on the front of the scanner and we made them believe that I was moving this tarantula, which was a Brazilian pink salmon bird-eating tarantula, as it's called. Pretty big. You see me picking it up there. That's my hand, so it's about the size of my hand. And what they would see me is randomly moving out of each of these different boxes this tarantula. This is actually an image of a subject in the MRI scanner. Okay, and they think that they can look down and see me moving this. And actually, what we did is we showed them the box with a spider in it, and then when we put them in the scanner, we did a lot of training on this, but we switched that then for videos, and we had about 20 subjects who believed that I was moving this tarantula in and out of the box. I was actually moving a fake tarantula in and out of the different box. Okay. The reason why we did that is because we didn't know how the tarantula was going to respond in the MRI scanner. And those of you, I actually have two tarantulas at home. But those of you who've worked with tarantulas, they can be a little bit erratic sometimes. And if they want to run, they can run pretty fast. So we didn't want subjects to be bit by a tarantula or become phobic to tarantulas because a tarantula runs up the scanner towards their face. Good thing about it as well is that we could decorrelate space and time. That was a bit difficult in the previous task. So I could do box 5, then box 1, and box 3, then back to box 1. So we decorrelated space and time. We're looking purely at spatial proximity here. So what we did then is we reorganized the conditions when we're doing the analysis. And what we're interested in here is looking parametrically how the brain activity increases when the tarantula is placed closer and closer to the subject's foot. And what we see here is that there's increased activity in arousal circuits, but also in really the peak here in the midbrain as the tarantula is placed closer and close to the subject's foot. I should say in the previous study, we did high resolution imaging, 1.5 millimeter. In this one, we didn't have that capacity to be able to do that. And this was run at Cambridge when I was a postdoc. When we look at the opposite effect, when the tarantulas moved away from the subject's foot, we see increased activity in the ventral medial prefrontal cortex, particularly in orbital medial prefrontal cortex. And we'll come back to this region in a little bit, because we argued at this point in time, in 2010, that this may be associated with this increased safety signal. Again, it's difficult to say what the cognitive processes are, but we believe that as the threats moved away from their foot, this might be a signal of increased safety. And the reason for that is because there's been some research out there showing that in spider-phobics who had undergone therapy, this was the one region that predicted their decreased phobia. The nice thing about these experiments as well is that you can reorganize the conditions, because they're so dynamic, to look at or ask different questions. So what we're interested in next was to look at how the subjects were monitoring over time the positions or movements of the tarantulas. So what we did, I'm going to give you a simple example here. We kept the position stable. So box 3, for example, which were box 2 and box 4 here. And what we did was we looked at the differences of when the tarantula would move to that same position, close to them or away from them. Okay, so we looked at the when they moved closer from the previous position compared to when it moved away from them. Okay, therefore, over longer periods of time, monitoring the movements or the danger of the threat as coming closer to the subject, while keeping the spatial proximity the same. And this is where we found the bed nucleus striatum terminalis came online, including the dorsal parts of the amygdala, which correspond to the central nucleus of the amygdala. So we argued here that at that time that this may be associated with monitoring or being vigilant towards, or over more sustained time periods, more vigilant towards the movements of the tarantula. So what we had shown here then in these studies was this sort of a set of parallel processes going on, that the brain at the same time is processing the spatial temporal properties of the threat, is keeping history of those, the threats, movements and proximity. And what I didn't talk about, it's making predictions about how much of a threat that spider is. And what we showed here is what we call expectancy error, is that when it was more scary than what they had predicted, that would be associated with increased left amygdala activity. I don't want to get any arguments about the left and right amygdala does, but that's where we found the core activity. But we didn't really talk about it in terms of laterality, and we still don't. But if we added them, we would have compared those two left and right directly. So the question is, though, with these original experiments, so we're looking at subjective ratings as they're going through the experiment, there was really no decision processes going on. So we went back to the behavioral ecology literature, and really one of the most used paradigms to study fear in nature, in animals, is a process known as flight initiation distance. And that was really made quite famous by this theoretical paper put forward by Yedenberg and Dill, okay, called The Economics of Fleeing from Predators. And it wasn't the first paper to talk about flight initiation distance, but it was the paper that kind of made it extremely popular. What is flight initiation distance? The distance at which prey will flee from an approaching predator. Okay. So that's sort of captured in this theoretical model here. And what we can see is that if you watch a David Attenborough BBC World documentary, you'll see that zebras, for example, will, live in herds. We've been studying herd behavior and safety in a couple of papers that are just about to come out. But they'll use the group as a safety refuge, okay? And then you'll see at the distance that they're typically always followed by a predator, okay? So what will happen is that our little prey here will stray away from the herd, maybe to reduce competition, to eat grass, for example, and feed. But there'll be a certain point that the lion here will get a little bit too close and begin to approach the prey, and the prey will flee back to the herd. And this is a mechanism that allows you to study not only the economic value of their behaviors, the value between being eaten by the predator and the value of eating themselves, but also threat sensitivity. And it's really captured here in this simple model. There's more complex models you can see behind. And again, you can read about this in the Nature Reviews paper from a couple of years ago now. What's happening is that the zebra is making two decisions, competing decisions against each other. The cost of not fleeing here in blue, that is associated with the increased cost is associated with being eaten by the predator, the increased danger of the predator. And the cost of fleeing is giving up density or giving up feeding behavior, for example. And there'd be an ideal point where if these costs of not fleeing and fleeing was identical, which we know is not true, but if they were, then there would be an ideal point, which would be the point at which the prey should flee from the predator. Of course, there's always going to be more emphasis on surviving predation than eating. So we propose then that there would be two types of fear that I mentioned briefly earlier. One which we call reactive fear, which is evoked when the organism is under attack. And reactive fear is a quick phasic coordinated reaction in response to an imminent threat that is or perceived to be directed towards the organism, and where there is little time to cognitively comprehend the danger of the situation. This is your fight and flight response, for example. Cognitive fear, again, this is when you're under attack, but now this is when the threat is more distant. Okay, you've got time to think before you react. Okay, and we refer to this as a conscious feeling of terror or horror, which results from the presence of a threat that is or perceived to be directed towards an organism, and where there is not only time to strategize escape, but also comprehend the forbidding nature of the situation. And our argument here is that when you're acting more on these reactive fear systems, you're going to be activating more of your midbrain paracaudal gray, okay, versus when the threat is more distant, you're going to be using more cortical systems to strategize your escape. Use your memory systems, for example, to predict when the threat will attack. So we create a very simple task. Is what we predicted, that when the subjects are encountering a very fast attacking threat, this will activate more of these fear circuits, okay? Particularly in the midbrain paracaudal gray. More cognitive or anxiety circuits, for example, will be evoked when the subject is encountering a slow attacking threat. So they've got more time to think about what the predator will do. So what did the paradigm look like? Well, first of all, the subject has to press a button to let this triangle there go back to this safety exit, okay? They can do that at any time, but the longer they wait, the more money they earn, okay? So that's the economic component to it. They're trying to make money while they're performing this task. They then encounter two different predators, actually three in the original experiment, but I'm going to simplify there. There was one with attack in the middle. But what they would do is that they would encounter a blue dot here that would attack them early, and it would attack any point within this Gaussian, okay? So there's a little bit of uncertainty there. Or they would be attacked by a slow attacking threat that would attack some point in this Gaussian here. So they're given time to make their decision. Now, to control for any confounds of timing here, we had matched control conditions, which I'll come to in a minute. But also, we always looked at 2 seconds before they made their decision, okay? So we're controlling for any timing issues here, because of course there was timing issues between these two different types of threat. Okay, so interestingly, looking at these different aspects of these fear, cognitive anxiety circuits and reactive fear circuits. And this was work done by Song Chi, who's now at NIH. If I can get off this slide. There we go. Okay, so what did we find? We found that when they were making this faster escape decision, okay, there was increased activity in the midbrain paracaudal gray, which you can see here. And you can see that quite nicely, that the increased activity here, but compared to matched control conditions for timing, okay, and compared to the slow attacking and medium attacking threat, there was a significant activity, not only the paracaudal gray, but also the mid-singular. And we argued this was more of this reactive fear circuitry. It may be associated with more sort of adaptive motor control, particularly here in the mid-singular, because we know these two regions are highly connected to each other in humans and in animals. For the opposite contrast, we wanted to look at what happens in the brain when they're encountering the slow attacking threat. And here we find that the ventral medial prefrontal cortex, including the frontal pole, were active, posterior, cingulate, and hippocampus. And again, we can see here, compared to the control conditions, these regions are significantly more active, and compared to the actual fast reactive systems, these regions were more active. So our argument then here was that that when you're encountering a slow attacking threat, you've got time to strategize. You're going to be using more of these cortical systems here, okay, of the hippocampus and ventral medial prefrontal cortex. But when you're not got time to really comprehend or strategize your escape, you're going to switch to these midbrain regions via these other circuits. Of course, brain imaging doesn't have the temporal resolution or spatial resolution to be able to fully dissociate these different parts of circuitry, but hopefully we will with some of the work that we're doing at the moment or plan to do. Okay, so as we mentioned, anxiety is associated with when there's a potential threat, or as we refer to as well as a distal threat, okay, going back to the original definition, and which is, we should see that when the threat is attacking us, is fast when it attacks us, we should be switching more to these fear circuits, okay? But when there's a slower attack, it should mimic more of these anxiety circuits. So our argument then was that trait anxiety would have an effect upon these slow cognitive escape decisions, but not these fast, fearful decisions, because they're in different circuits. So what did we find? We found that trait anxiety was correlated with the subject's flight decision, but only for the slow attacking threat, when they've got time to comprehend that situation, okay? But when they're making a faster decision, which mimics more of a fear response, we found that that had no correlation with their I should say, their flight distance, the distance in which they fled. So what you're showing here then on the left is that our anxious individuals fled earlier, okay, than low anxious individuals, but there was no effect for the fast. And again, here what we've done is we've controlled for the variance. We've also added in the mid condition to control for any spatial differences, and we again find no differences between the mid if you combine the mid and the fast attacking threat. It's only for this slow attacking threat that you see that correlates with trait anxiety. What do we see in the brain? Same thing. For the fast attacking threat, trait anxiety correlates with nothing. For the slow attacking threat, we see there's a strong correlation between hippocampus and the ventral medial prefrontal cortex, as well as the mid-cingulate and insula. Okay, so the higher you score on trait anxiety, the more you activate these regions of this, what we refer to this sort of cognitive fear or anxiety circuitry. And this really fits nicely with the work by Josh Gordon and others that are showing these same circuits associated with anxiety. We then run a PPI between these two regions, the hippocampus and ventral medial, and the strength of the connectivity between these two regions is increased as the trait anxiety increases as well. Okay, so we wanted to, in this last study, which is not published yet, is under review, we wanted to tap into these hippocampal and prefrontal regions a bit more, okay? And again, we went back to very limited, there's only a few papers on this, but very limited research from the field of behavioral ecology to use this concept of spatial margin of safety decisions, okay? And we wanted to look at how close the subjects will place themselves to safety under uncertain or more predictable attacking threats. Again, this is work done by Song Chi. So what is margin of safety? It's the distance in which prey will move from a safety refuge, okay? And what you find in some of the limited behavioral ecology literature is that, a prey, for example, a rabbit ear, will stay closer to a safety refuge under conditions where it's either an high density predator environment or more of a volatile predator environment. So they'll stay closer when they can't predict where or if they're going to encounter a predator. But when they know the environment, it's a more predictable environment, the prey will move further away from the safety refuge. Again, there's other interconnected circuits that would go on there in terms of feeding milk mating behavior that would change this. But generally, keeping everything equal, you'll see this difference between how far the prey will move from its safety refuge, which we refer to as margin of safety. Create a very simple task here. Subjects are given the contingencies: high reward, low reward, high shocks, low shocks, and then they encounter one of these three spheres here. Okay, what are these three spheres? They're the different types of predators that they will encounter. Okay, so if we move over to C over here, what we can see is that red represents what's known as a leptokurtic attacking threat. Okay, this is more of an uncertain threat. So a leptokurtic distribution here is a distribution that contains more outliers. It's more difficult to predict. Okay, the lepto means skinny, so you can see it's much more skinny at the peak here of the distribution. Okay, and this has been used in economics as a way of inducing uncertainty in decision making. We then add two normal distribution Gaussians, one that was matched for the variance, okay, which you can see here in green, and one that had the half variance. Now, argument here is that the half variance is the one that's easiest to predict. The matched is the next difficulty in terms of prediction, and then the leptokurtic is the one that's most difficult to predict when or where it will attack. That has to rate their confidence of escape. And then they do this for 10 rounds. Okay, we're trying to get as many of these conditions and get them to learn them as best possible. So we clump them together into 10 rounds, and they would continue to do this. And actually this task was 2 hours per subject. They would do it over 2 sessions over 2 days, 1 hour one day, 1 hour another day, counterbalanced. So, and this was optimized for multivariate pattern analysis, which I'll come to in a minute. So the subject has to either place them, they're shown the virtual predator, and they have to learn where to place themselves closer or further away from safety. If they place themselves further away, they get more money. Okay, they earn more money. It's the economic component to the task. But if they place themselves closer, then they're more likely to escape. And I should always say, in all of these tasks, if they get caught, they're not only receiving an electric shock, but they also lose the money as well. Okay. So we do that. They make the decision for 4 seconds, and then they can't do anything during that phase. But that's followed by a 3-second screen where they actually move the cursor to the position, because we want to control for any motor confounds in this experiment. As you know, with MVPA, you have to really keep the conditions as identical as possible. You can't control for such things as action planning, for example, but we could control for the motor movements, which we did here. Then what happens after they've placed themselves closer or further away, they see the outcome. So they learn where the threat attacked them. They just see the outcome. They don't see it oscillate towards them like they did in the previous flight initiation distance task. They just learn over time the spatial distributions of these attacking threats. Okay. And then they find out if they won or lost the money. So what do we find? That during the margin of safety, when they place themselves closer or further away from the threat, you find that that for the leptokurtic, the more unpredictable threat, they place themselves closer to the safety exit. Okay. We found no differences though between the two Gaussians. Okay. The half matched and the matched Gaussian variance, matched variance Gaussian. Okay. And again, for the confidence of escape, we found that they were less confident of escaping the leptokurtic one, which is just proof of concept there, but really no differences between these two Gaussians, which was good because we originally weren't going to add the leptokurtic. We just thought the variance may change it, but we would have got a nil result if we'd have done that. So next thing we want to do is look at what's going on in the brain when they're making these decisions. So again, remember, we're using MVPA, we're looking purely at that decision, the margin of safety choice that they're making, okay, control for any motor issues. The only thing that's different is the context of the threat. Is it one that's predictable or not? But what we're looking at here is basically just the decision across all of the trials, okay? So we use a support vector machine here, leave one out approach using PyMVPA, a standard technique. And again, what we're not doing here is we're not separating whether they make a safe or dangerous decision. We're just looking purely at their decision. Okay. So what did we find? We found using the MVPA that a series of regions came up in these, particularly these regions that we discussed before, the hippocampus and the ventral medial prefrontal cortex. And we thought this was really, really fascinating because we'd had this prediction that if they're making a safe decision, they're going to activate the anterior. If you remember, we talked a bit about this before in the tarantula study, and we actually followed that up with several studies showing that when subjects are given a safety cue, it seems to activate this region of the anterior ventral medial prefrontal cortex. But when it's a danger cue, there's increased activity of the posterior parts of the ventral medial prefrontal cortex. We then looked closer. We found there was no difference across all these different conditions in the hippocampus, whether it was predictable or not, But we found that when they were encountering the more leptokurtic, the more difficult and predictable threat, it activated more or was associated with the posterior parts of the prefrontal cortex, where the other two Gaussians did not reach threshold for significance. Okay, so this seems to be driven by the more leptokurtic threat, more unpredictable. However, for the easiest one, the one that was very predictable, we found that this seemed to be driven more by this. That was our speculation, but we want to tap into that a little bit further. We did a meta-analysis in a paper that's just about to come out in ticks, looking at limited, but there's quite a few papers building up now looking at safety signals in the brain and threat signals. And we found that each of these represents a peak from a study. We found that there's this difference between these anterior and posterior regions associated with threat signals and safety signals, okay? Just to put that into context. So we want to follow this up. We usually ran a univariate analysis, and we wanted to run a PPI to look at coupling between brain regions. There's arguments whether that means connectivity or not, but it certainly does mean that there's coupling between these brain regions due to their similarity in their time series. And we found that for the leptokurtic unpredictable threat, there was connectivity, significant connectivity that went to hippocampus and the amygdala. When we put the seed in that peak from the MVPA that we just mentioned or just discussed, we found that there was increased connectivity to the striatum for both Gaussians that were predictable. We then ran a parametric about how close they placed themselves to safety or how further they moved from safety. When they were, those who moved themselves further from safety during the dangerous condition, there was increased in this more posterior region. And when they placed themselves closer to safety during the safe condition, there was increased for both the Gaussians in the anterior parts of the ventral medial prefrontal cortex. So our argument was that predictable threat will activate these different cortical systems, okay, and have different connectivity. We found, as you can see up here, though, that the dorsal medial seem to integrate all of these or have connectivity with all three different types of threat. We also ran a simple reinforcement learning model, and when we looked at our prediction errors on the task, it seemed to activate these two regions, again, the hippocampus, amygdala, and the striatum. But I'm not going to go into that due to time. Okay, so summing up here, I want to talk about or mention that fear and anxiety are a dynamic process that involve a complex set of defensive circuits. And distal or slow attacking gradual threats via these cognitive fear circuitries overlap with the anxiety circuitry, which includes the ventral medial prefrontal cortex, hippocampus, posterior cingulate, and amygdala. Proximal, imminent, fast attacking threats seems to be involved in more motor reactions and also the midbrain paraconductal gray, and we've shown these now over several studies. Avoidance decisions that we saw in the margin of safety also seem to activate these circuits, hippocampal prefrontal circuits, again, that we discussed in the context of prospection and memory and anxiety. And one sort of argument here, sorry for spelling mistake there, is that the higher order representation fear may be involved in the frontal pole. This really goes back to the work of Joe Ledoux, his theoretical work at least. So again, I want to thank all these individuals and my lab at Caltech who do all of the hard work here. I'll just get to sit back and do these wonderful presentations. Thank you.
Thank you very much, Professor Mobs, for this great talk. And I'm just sorry I couldn't unmute myself during the hitting part of the tiger, but let me reassure you, the audience probably felt the same. We have a first
Yeah,
question. I think it relates to the first part of your talk. How does
yeah.
PTSD change the defensive circuits you talked about? If you could.
Yeah, it's a good question. I've always wanted to run this on PTSD, but we're running these on general anxiety disorder. I can tell you a little bit about that with Michelle Krass, but we've not had access to PTSD patients, and we'd love to do that research. What we think is that, in particularly, that you're going to mirror these types of responses you can see here. That PTSD is going to result in more threat sensitivity to these different responses. They're going to flee earlier. They're going to place themselves close to safety. What we also believe that and this is where the decision component really came, which was quite nice in our flight initiation distance paradigm here. And again, what you see is that our argument here is that when you're you're fleeing earlier from the threat, you're switching between these different fear states. You're going from more of this sort of system of cognitive fear where you're controlling your responses and you're switching more over to these reactive systems. And we couldn't show that in the original experiments, the tarantula and the Pac-Man type experiment. But our argument back then is that, and in the 2015 review actually with Adam Perkins in ticks as well, the sort of argument there was that you'll see this this switch will occur earlier. Okay, and that's what we tried to get to in some respects, if I can get over here. In the model, in the recent TICS paper, where we talked about these transition states here. And what we believe is that although you'll see increased arousal in someone with PTSD or general anxiety and so on in these, what you're going to see is that people who have PTSD, and this is my theory, you guys probably know a lot more about than me, they're just going to switch it. They're going to switch earlier over into an higher level defensive state. They may even skip these states. They may go from post-encounter or even from safe and switch directly over to panic, for example. So we think about this as a continuum, but you can skip these states in pathological conditions, I believe. To answer that question?
I believe so. Thank you. And just a follow up, how clear was the cut in the healthy population you looked at between the anxiety phenomenon and the fear manifestation you looked at? Could you tell like several groups with different properties, or was it everyone more or less had a clear border?
Well, we're looking at correlation there. And I think that what we're looking at is that we only had like 29 subjects in that experiment. And we would have added more, but I did the study at Columbia and then I left and we did the analysis at Caltech. So we'd have had to start the study completely over again to get more numbers. But what we're doing now is we've got a new version of our threat image continuum, which I didn't present. And we have a shorter version of these tasks, of the FID task, flight initiation distance task. And we're running that on very large samples of up to 200 subjects. So it's more of a sort of local, we use it also more like a localizer for every single other study that we're continuing to do. Because then what we can do, and hopefully with grants we have in and look at these different populations over time, we can begin to separate them in a more fine-grained way and look at that separation between the different groups. But of course, with a sample of 29 on a correlation, you're really limited with that sample size, but it's something that we'd love to do. And we're doing, but not with clinical samples yet because we don't have all the different ones. But we're certainly going to be running this with depressed and general anxiety and then and potentially panic disorder.
Right. If there are any other questions, please feel free. Okay, so just a second. So Dr. Maria Modi asks, sorry if I missed this, but do you distinguish between real and phobic type fear-related activations?
Yeah, we didn't look at that because that tarantula study originally got turned down, not surprisingly, by the IRB in England. and I some restrictions on me in terms of what I could do with the experiment, and that's the way it turned out. Although we were encouraged later on to run this on phobic subjects, and again I left and moved to New York, so this transition thing, and when you move somewhere, you just start to do new stuff. But we do now have tarantulas, and we have a new set of experiments with tarantulas. And the first thing to do is to run these experiments on a larger healthy population, and then run it hopefully on phobic. I think it might be a little bit difficult where we are because Caltech doesn't actually have a medical school, so we'd probably have to ship it off and do it somewhere else. But we have close links with other places, so that's a possibility. But it's a good question. I'd love to answer that question. Because I study basic science, I don't study clinical populations. I'm not a clinician, but from a theoretical perspective, I'm interested in that to see how you can, how plastic or you can push and pull these different systems based upon, pathology and so on.
Thank you very much. So Professor Mobs, thank you very much again for this great talk, and we hope to see you in person, hopefully here, but maybe even in California one day.
Yeah. Great. Thank you.
Thank you, everybody.